All right, first I want to thank John and Francisco and Michel for inviting me in the honor of this presentation today. Thank you very much. I also want to thank you for suggesting the title of my talk, which is Clonality, Evolutionary Curse or Blessing. And, oh, thanks. And so the way I approach this topic is from the perspective of Darwinian fitness. So what, in terms of Darwinian fitness, the transmission of genes from one generation to the next is, to what extent is clonality an evolution, a curse or a blessing? And I should, one more bit of information to set the context for this talk, and that is that, like Spencer Barrett in yesterday's talk, I'm a plant reproductive biologist. So I'm going to be borrowing quite heavily from the terminology and concepts that Spencer Barrett gave yesterday. So it's a bit of a departure from the talks that we've been having in the rest of the afternoon session. And one of those key concepts was, Spencer gave a quote from John Harper, a very influential plant ecologist 40, 50 years ago, and that is that clonality is just growth. Fitness, sexual fitness comes through sex. And I'm going to adopt that perspective in this talk today. Clonality impacts fitness via its effects on sexual reproduction. And so, and Spencer told us the main mechanism, the main curse in the context of this title of how clonality might affect sexual fitness. And that is through this concept of mating interference. And basically, as clones get bigger and bigger and bigger, the probability that gametes will be transferred within the clone rather than being transmitted to other clones, enabling outcrossing, increases, and that causes mating interference. It's thought to be a major source of major fitness disadvantage to clonal organisms. I want to come to, at the end, I want to come to this idea come back to the idea of blessings, and I want to tell you about a result that's emerged from some modeling we've done recently that provides a new benefit, potential benefit, or at least one that's not been described in terms of, in the plant literature of clonality in terms of Darwinian fitness, and that is that clonal expansion can promote the effective dispersal of seeds because of the way clones spread through space. Okay, along the way, I'll tell you about the details of that modeling effort, or some of the details, and I'll preface that by considering how clonality affects fitness in slightly more detail, starting with this idea of mating interference. But just to set the context a little more to sort of so you can see where I'm coming from on this topic, I'm just going to show you some of the organisms that I've studied that have informed my thinking on this topic. So the very first plant I studied is this woodland clonal herb here, Clintonia borealis. I studied patterns of self-sterility among clones in this plant. So the next plant I studied was this plant here, which is one that Spencer mentioned yesterday. And here I studied the evolutionary loss of sex via the accumulation of sterility mutations. And just to keep it rather short, the basic idea here is that at the northern periphery of the species range of this plant, the sterility mutations have accumulated, different sterility mutations in different populations at this northern periphery. And these sterility mutations have a selective advantage in these harsher environmental conditions. They seem to have pleiotropic effects on the survival of these clonal genotypes at these northern populations. But in terms of the transmission of genes through sexual function, that's not a blessing of clonality that I'm going to consider in this talk today. So I'm not going to talk about this plant again. One plant I will talk about again, however, is this plant here, Sagittaria latifolia. This is the plant that I've done much of my research on, including the research that I did in Spencer Barrett's lab. I was a PhD student of Spencer Barrett's over a decade ago. And I'll talk a bit more about this plant in an upcoming slide, so I'll just skip over that right now. Now, all these plants are highly clonal, and that's not a coincidence. I mean, I am interested in clonality, but if anybody who's studying a herbaceous perennial plant in mid to high latitudes, chances are good that the plant that you study is going to have some capacity for clonal growth. Clonality is pervasive. The vast majority of plants, particularly perennial herbaceous plants, have some capacity for clonal reproduction, and the proportion of the vegetation that is comprised of clonal plants can reach very high numbers up to 90% in some ecosystems. And clonality is associated with many benefits to the vegetative performance of plants. And Spencer mentioned quite a lot of those in his lecture yesterday. I'm not going to talk about them at all in any detail. And vegetative performance is one thing, but in terms of the Darwinian fitness, and if we think about that in terms of the transmission of genes via sex, these benefits that accrue to clonal organisms via vegetative performance, their ultimate effect on fitness gets filtered through how they affect the sexual performance of these clonal organisms. And that's what I want to consider now. So the literature on the topic of how clonality affects reproductive success, sexual fitness in clonal organisms probably started, the literature started with this paper here by Stephen Handel, in which he considered, well, I'll just read the title. He called it The Intrusion of Clonal Growth Patterns on Plant Breeding Systems. And the title sort of speaks for itself. The idea is that the increased size of clones has this byproduct in terms of sexual fitness. As the clone gets bigger and bigger, the chances are the pollen is going to be exchanged within the clone, and that is going to cause a lot of selfing and what we've since come to call mating interference within the clone. It can affect the fitness of the organism through its seed function because those seeds are highly inbred. It can also reduce the function of the plant through its pollen because that pollen is, rather than being involved in outcrossing, it's being involved in selfing. And there's much more, as Spencer mentioned yesterday, much more efficient ways of going about selfing rather than having it transferred among the different modules that comprise a clone. So, and this idea of mating interference has been pervasive in the literature. Every review paper that's come out since then has really focused on this concept of mating interference in clonal plants. There's been a few through the years, including one that Spencer and I were co-authors on, in which we, again, picked up on this idea of mating interference in clonal plants. And so the idea is that, as I've already said and Spencer mentioned yesterday, is that as clones get bigger, they're more likely to suffer from interference during mating, and that this has major fitness consequences for fitness through male and female function. Now, that's sort of where the field is at the moment. What I want to do now is take a step back and say, okay, well, clonality is just one way that plants have of getting big. And is this mating interference something that's intrinsic to clonal organisms, or is it just intrinsic to becoming larger in size? And I'm going to make the case that this mating interference is something that's intrinsic to an increase in size, and that clonality actually reduces mating interference in comparison with other mechanisms that an organism might have of increasing in size. And then I want to consider other potential benefits of clonality that come from aspects of the life history of clonal organisms that haven't really been included in this discussion of sexual fitness in clonal organisms, and that occurs during the dispersal of seeds. The focus has always been on mating, events that take place during mating. What I want to talk about at the end is events that take place after mating, which are just as important in terms of determining the fitness of these organisms. Okay, so we're now into the second stage of my talk, and I want to consider the various aspects of clonal organisms that affect its sexual fitness. And the most direct way that clonality can affect the sexual fitness of an organism is via trade-offs. If there's a fixed resource pool that an organism can allocate to different activities of its life history, clonality or sexual reproduction, then any resources that are devoted away from sexual reproduction obviously can reduce its Darwinian fitness. Okay, so there's quite a bit of literature on trade-offs in clonal plants, but most of them have focused on this rammet level. So this is this term that Spencer mentioned yesterday. These are the individual shoots, these individual modules that comprise a clone. Now, that's okay, but there are plenty of good reasons to suspect that these trade-offs do not scale up to affect the fitness of entire clones. Okay, so what about studies that have studied trade-offs that occur at the level of entire clones? Well, the data there's only been a couple studies, and the data from those studies is not entirely clear as to whether those trade-offs occur or not. So the first thing I want to tell you about is a study that I did to investigate or that was done in my lab by a graduate student of mine, I should say, to investigate trade-offs, resource-based trade-offs between sexuality and clonality at the genet level. And we used this plant here, Sagittaria latifolia. It's a plant that Spencer mentioned yesterday. He showed a very similar slide. So the plant is quite interesting. It's very unusual among flowering plants in that populations can be monoecious hermaphrodites. So they have these unisexual flowers that are female and male, and you have populations of hermaphrodites. Or you can have populations that are entirely female or entirely male. And one of the things that I did when I was a PhD student in Spencer's lab is work out the genetics of the system. It's simple Mendelian genetics with XY sex determination, or effectively XY sex determination in these dioecious populations. Okay, so the nice thing from the perspective of this experiment I'm going to tell you about, the nice thing about using Sagittaria is that it has unisexual flowers. So you can independently manipulate patterns of investment in male versus female sex function. The other thing, and the more important thing that's nice about working with this organism, is that at the end of the growing season, there's only 2 bits of the plant are left. The individual shoots are ephemeral. They die back at the end of every growing season. And at the end, there's only 2 things left. There are seeds that are the product of sexual reproduction, and there are these corms that are the vehicle of clonal propagation. So we have 2 bits of biomass that are left at the end of the growing season, and so we can use biomass as a common currency to evaluate trade-offs between sexual investment in sexual function on the one hand and investment in clonality on the other hand. And that's what my student Wendy Van Drunen did with Sagittaria latifolia. We took plants from a bunch of different dioecious and monoecious populations, and we subjected them to 2 different, in the dioecious side at least, 2 different treatments, which were, so we took groups of females and males, and we removed all the flowers, and we did this at the whole genet level. So we let these plants clone up, and then we applied these treatments to entire clones. We removed all the flowers, so we prevented the investment in sexual reproduction at a very early stage of development. Or we let them reproduce sexually normally. And then from the monoecious populations, or plants from monoecious populations, we did the same things. We prevented them from reproducing sexually by removing all the flowers at an early stage of development. We allowed them to reproduce normally, or we just removed one class of flowers or the other class of flowers. So here we would remove all the male flowers and we'd have females, or remove all the female flowers and we'd have males. And we did this to a bunch of different plants in a large common garden experiment in a field right beside my office. And that's Wendy's head just poking out. She's doing the manipulations on these clones in the common garden. And so the results were quite clear. So we have our 3 different sexes here at the bottom, and the amount of resources, biomass that they invested in clonal reproduction on the y-axis here. Let's just focus on these females for the moment. If you prevent females from having sex, they have much higher investment in clonality than plants that did engage in sexual reproduction. So I guess one thing that might have jumped out at you already is that males don't show this pattern at all, which suggests, so here it suggests that females are paying a cost in terms of the clonal propagation, in terms of reproducing sexually. Males do not appear to be paying this cost by this metric. I don't want to give you the impression that investment in male function has no cost. It's just that males pay their costs in a different currency. So males that reproduce sexually have much lower nitrogen content in these corms than males that didn't reproduce sexually, about a 4-fold difference. So a massive reduction in nitrogen content, and of course nitrogen is a very important nutrient that limits plant growth in plants. So males are paying a large cost in reproduction too, but just in a different currency. But I'm just going to focus on this currency here, biomass. So females are paying a large cost. There's a big trade-off between investment in clonality and sexual reproduction. Hermaphrodites are showing the parallel response. And the interesting thing from these data was that if you do a full accounting, that the biomass that females lose in terms of their investment in clonality is exactly made up for the biomass that they gain in terms of the production of seeds. So all the biomass that these females used in seed didn't use all the biomass that these females didn't use in seed production went to the production of corms instead. So you have this one-to-one trade-off between the investment in sexual reproduction versus clonal propagation, at least by this metric in this plant. So we have this very clear trade-off that scales up to the whole genet level in this plant. Okay, so plants invest in sexual reproduction, but then how does that affect fitness? So here I want to turn to the gain curve concept that was popularized by Eric Charnoff back in the late 70s and early 80s. And the gain curve concept just describes how investment in sex functions determines Darwinian fitness. And the pattern that we expect to get for the flowering plants is this one. We expect to get this type of relationship between investment in sex and fitness. I'll explain why in a moment, but this is the most commonly expected pattern in terms of how investment in sexual reproduction should scale with fitness. There are mechanisms by which plants can linearize their fitness gain curves, such that there is a direct correspondence between investment in sexual reproduction and fitness. And even less commonly than that, there has been some speculation in the literature that you could have accelerating relationship between investment in sexual reproduction and fitness. But as I said, what we generally expect for plants is that you get these diminishing returns on investment in sexual reproduction in terms of the fitness payoff. So why does that occur? The reason is that the dispersal of pollen and seeds is highly spatially restricted in populations of clonal plants. We find this for any type of plant, wind-pollinated, animal-pollinated, wind-dispersed seeds, animal-dispersed seeds. We tend to find this pattern here where the vast majority of seeds or pollen are dispersed very close to the source plant, and very few pollen grains or seeds are involved in longer distance dispersal. We have this leptokurtic dispersal of pollen and seeds from a wide variety of plants and animals. So how does that then cause the gain curves to diminish like this? Well, what happens is that as plants invest more and more in the production of seeds or pollen, so they're investing more and more in sexual reproduction, those pollen grains are being dispersed close to the parent plant. They end up competing with one another rather than being dispersed far enough away that they're not competing with one another for siring opportunities. Same thing happens with seeds. So the more and more seeds a plant produces, the more and more seeds that are deposited around that parent plant, and they end up competing with one another during seedling establishment and germination. So in general, we expect the gain curves to diminish with the patterns of investment in sexual reproduction. Okay, so we have these two concepts, trade-offs, and these gain curves. How do we then, I want to put these together and compare non-clonal and clonal organisms and how these things might play out in terms of the costs and benefits of clonality in these systems. Okay, so here, I know it looks a bit blurry. It's meant to look blurry. We're looking down, the idea here is that we're looking down on a patch of plants, in this case a non-clonal patch of plants, that are dispersing seeds and pollen from a point source. So this is the non-clonal situation. And because pollen and seeds are being dispersed from a point source, and because most of that pollen seeds will end up very close to the source, those pollen grains will end up competing with one another for siring opportunities on other plants. So pollen from this green plant will end up competing with other pollen grains from this green plant for siring opportunities on this red genotype, or for siring opportunities on this blue genotype. So much of the pollen produced by this green genotype will end up being wasted through these competitive interactions. Same thing occurs during seed dispersal. A lot of the seeds will be dispersed to locations very close to the location of the source plant, and will end up competing with one another during establishment. Okay, so this is the non-clonal situation. What about the clonal situation? So if plants subdivide their reproductive function among these spatially separated modules in the population, plants now have, each individual shoot might have a smaller investment in reproduction because of trade-offs, but they've increased the number of potential, they're now near neighbors. So this green genotype now has near neighbors represented by all the different genotypes in this patch. So it's near neighbors to brown and red and green, not just blue and red as in the previous slide. And moreover, it's increased its access to different mating partners. Moreover, each of these plants is dispersing fewer seeds than in the previous scenario because of trade-offs. So there'll be fewer seeds competing with each other for establishment opportunities in the next generation. Okay. So these are the ideas that have sort of been bubbling away, and started putting some of them together in this in the review article that I mentioned that was co-authored with Spencer Barrett about 5 years ago now. And so the first model that we came up with was a graphical model in which we tried to put some of these ideas together. Okay, so here on the x-axis we have this gain curve concept, but at the individual ramete level. So we have the amount of resources invested in sexual reproduction by an individual shoot within a clone, and then the contribution of that individual shoot to the fitness of the genotype. Okay, and so an individual ramete could invest a large proportion of its resources into sexual reproduction, or relatively little. And the fitness payoff, or the marginal fitness gains from that investment, is defined by the tangent of the slope of the tangent to the curve at these different locations. High levels of investment in sexual reproduction here, low levels of sexual reproduction over here. Okay, so although total fitness is lower, the marginal fitness gains are higher at lower levels of investment in sexual reproduction for this red scenario versus the blue scenario on the right. Now, if there are trade-offs, and now we know that there are trade-offs between investment in clonality and sexual reproduction, then plants that are investing a lot, or rametes that are investing a lot in sex, will have fewer resources left over for the expansion of the clone. Whereas these plants will have more resources left over for the expansion of the clone. So what we expect then is that those rametes that invest a lot in sexual reproduction will find themselves in clones that are small, are clones that have a small number of rametes. And rametes that are in clones that invest little in sex per ramete will find themselves in clones that consist of a larger number of rametes. So you put those two things together then, and you think about how the investment in sex per ramete might then scale up to affect genet fitness. We get this sort of pattern here where if the per ramete investment in sex is quite low, we expect larger overall fitness than in the reverse, because of these effects of gain curves and trade-offs that I mentioned in previous slides. Okay, but this is just a graphical, this is a conceptual model. Does it hold any water? So we tried to make this a little more rigorous by doing some mathematical modeling. I'm not going to show you the details of the math because I will show you, there's a more up-to-date model I want to show you in a little bit, and I'll show you some expressions from that model in a moment. But basically, we go back to this idea of gain curves in clonal organisms. And and here we're just focusing on investment in just one half of, or sex, right? So we're talking about investment in male function. That was the focus of this particular paper here. That's a detail. So here we have investment in sex on the x-axis and the fitness payoff from that investment in sex. For a clone made up of one ramete, so a non-clonal plant. As the next sets of curves just show the relationship, the shapes of the gain curves that we expect for different sizes of clones based on these principles that I was telling you about before, trade-offs, and how the subdivision of reproductive effort among clones might affect the fitness payoff for clones of different sizes. So as clones get bigger and bigger, the slope of these fitness gain curves increases. This is an absolute measure of fitness. So fitness peaks at intermediate levels of investment, or at intermediate clone sizes in this model. Okay, so the problem with this model and all the other models that have looked at this idea of how clonality might affect sexual fitness is that they're not spatially explicit. And of course, spatial relations among plants are one of the key biological aspects of clonal organisms. So because of the effects of dispersal and so on that I've been telling you about. So one thing that was missing from all these other modeling attempts was that they weren't spatially explicit. And so that's one thing that we needed to include, and that's what I want to tell you about in a moment. So I think the key elements of a model that really tries to come to grips with the curse or blessing of clonality in terms of Darwinian fitness has to have at least these three elements. It has to invoke these trade-offs, be spatially explicit, and include the dispersal of pollen, so evaluate the effects of this mating interference that has been the focus in the literature so far, but also factor in what's happening during seed dispersal, because that's the other half of fitness. All this mating interference is one thing, but ultimately these seeds need to be dispersed and become established, and that's going to be a major bottleneck of fitness. Okay, so I just want to go through some of the conceptual details of the model without showing too many expressions. Okay, so we assumed that there were trade-offs in the model. We assumed that organisms had a fixed budget for investment in clonal versus sexual reproduction. So this is sort of the Sagittaria latifolia model of a plant. At the end of a season, there's just two things left. There's the clonal, there's the product of clonal reproduction, and there's the products of sexual reproduction. So we have investment in clonality, this variable g here, and then the investment in sexual reproduction, which is divided into its female and male components. This, of course, determines the production in seeds, but those seeds need to be produced following pollination, so we included both male and female investment in sexual reproduction. And to keep things relatively simple, because there's a lot of other complexities in the model, we assume that the allocations to male and female function were the same. So then clone size we just assumed was a linear function of the investment in clonal growth, G. And then reproduction was then subdivided across all the different rametes in the clone. So across all the N rametes in the clone. And that investment in male and female function were equal and scaled by the amount of resources left after the investment in clonal reproduction had been made. Okay, the model was spatially explicit in two ways. One way was that we defined the spatial relations among the different genotypes in the model. And we used this framework that Spencer mentioned yesterday, this phalanx versus guerrilla concept of clonal growth strategies. On the one hand, we have phalanx strategies where plants grow, clones expand at high densities. There's no intermingling of different clonal genotypes. And so clones are not intermingled under phalanx growth strategies. Under guerrilla growth strategies, clones spread out, they can fragment or have long stolons, and the clones become highly intermingled under guerrilla strategies. And there are also intermediate growth forms. Spencer mentioned a whole bunch of different mechanisms by which these different strategies can arise in clonal plants, and that's a detail that I don't want to get into right now, in part because Spencer mentioned it yesterday. So it's a continuum, and the extremes are these phalanx and guerrilla. We considered three strategies. We considered a phalanx strategy where there was very tight clumping of clonal rametes, a guerrilla strategy where there was lots of intermingling, and then an intermediate strategy which was just exactly in between these two extremes in our model. Okay, so here is a little more detail on how that was done. We had two spatial parameters here, B and R. R is the distance between different genotypes in the model, and B is the maximum distance between the rametes in a clone. So in the phalanx strategy, B was 1/10 of R. So the different rametes in the clone were clumped together and did not intermingle with other genotypes in the simulation. It's not a simple. Well, we did simulations, so we did two models. We did an invasion analysis, more of a mathematical analysis. It wasn't amenable to formal solution, so formal analytical solution because of the complexities involved in the dispersal function. So we used pairwise invasion analysis. This is a type of evolutionarily stable strategy analysis that some of you may be familiar with, in which you have a resident and a mutant strategy, and at equilibrium, the resident and the mutant strategies are the same. In this particular depiction, I've shown a mutant clonal phenotype and a resonant clonal phenotype, but as I said, at equilibrium, those phenotypes are the same. So here's what a gorilla strategy looked like in the model. The maximum value of B that we considered was 5 times the intergenet distance. So the maximum distance that a clone could be separated was 5 times the average, well, the distance between different genotypes in the population. So this is a gorilla strategy. We also considered an intermediate strategy. We also had spatially explicit dispersal in the model. So we assumed that pollen and seeds were dispersed following a Gaussian dispersal kernel. So this is the pollen dispersal kernel and the seed dispersal kernel. To keep things relatively simple, we assumed that those two were the same. So that DP is the average dispersal distance and DS of pollen, and DS is the average dispersal distance of seeds. We assumed, to keep things relatively simple, that those distances were the same in the runs that I'm going to tell you about. And so here is just a depiction of what a gorilla strategist competing with a non-clonal phenotype might have looked like in terms of the dispersal of seeds, the spatial dispersal of seeds and pollen in the invasion analysis. And likewise for a phalanx strategist. So here's what would happen: the dispersal of seeds and pollen from a plant from the different ramets of a clone with a phalanx strategy. Okay, selfing. So a key element we wanted to evaluate was this idea of mating interference. And so we allowed selfing to emerge from the properties of the model. So selfing rates were had 2 components: pollen dispersal within ramets and pollen dispersal among ramets. And so pollen dispersal within ramets was just the dispersal of pollen at the point source. And pollen dispersal among ramets was just the sum of the pollen exchange between ramets, which were spaced B units apart. So we didn't set a priori selfing rates, which is an important feature of this model. Okay, what we were trying to evaluate then was the equilibrium value of the investment in clonal growth. So can clonality be favored under these different scenarios which we've considered so far: gorilla versus phalanx strategies, different dispersal distances, different magnitudes of inbreeding depression, and different magnitudes of the spatial separation of ramets. So this phalanx versus gorilla form of clonal growth. So now I just want to tell you about the results of this modeling exercise. I should say that we did stochastic simulations as well, and the results complement those. I'm just going to focus on the results from the invasion analysis. Okay, so here's this key result here, or the key thing that we were interested in, I should say, the equilibrium value of G. This is the investment in clonal growth for a given set of dispersal parameters, of magnitude of inbreeding depression, and the degree of clonal intermingling. And a few things are readily apparent. As the degree of clonal intermingling increases, the investment in clonal growth increases as well, particularly under dispersal limitation, this black line here. So this is spatially restricted dispersal. The red line is intermediate dispersal distances. The blue line here is broad dispersal distances. Clonality is only favored if this pollen dispersal and pollen and seed dispersal are broad, if clones are highly intermingled. So this is in the absence of inbreeding depression. We considered various values of inbreeding depression. I'm just going to show you the 2 opposite extremes. This is complete inbreeding depression. Self-seeds are fully inviable. One of the perhaps surprising things that came out of this modeling exercise was that higher magnitudes of inbreeding depression actually promote the investment in clonal growth. So higher inbreeding depression favors clonal growth, as does dispersal limitation, which I've already mentioned. Okay, so the obvious result here is that as clonal intermingling increases, so does the equilibrium investment in G. This is something that is totally obvious. Spencer mentioned this yesterday. Without the benefit of going through a modeling exercise, this is obvious to anyone who's thought about this problem at all. So this is not a particularly interesting result. The interesting results come out of the effects of dispersal limitation. So the fact that clonality appears to be most strongly favored under dispersal limitation suggests that clonality might be a strategy to avoid dispersal limitation in the flowering plants. Remember, I told you that pollen and seed tend to be the dispersal of pollen and seed tend to be highly spatially restricted. Clonality might help promote the spatial dispersal of pollen and seeds. The other perhaps surprising thing is that we had higher levels of investment in clonality under strong inbreeding depression than in the absence of inbreeding depression. This is because mating interference is actually weaker in clonal organisms than in non-clonal organisms. And this is the point I was trying to make earlier. So this curse of clonality, this mating interference, is actually a weaker force in clonal organisms than in non-clonal organisms. And this is because selfing rates decline with clone size in this model. So this is the allocation of clonal growth here, so which defines the size of the clone. And on the y-axis here is the proportion of self-seeds. And you can see for different dispersal distances, the selfing rate declines. Of course, it declines only weakly when pollen and seeds are dispersed over very broad distances, but it declines very strongly when pollen and seed dispersal is spatially restricted. So clonality appears to alleviate mating interference rather than promote it, as has commonly been assumed in the literature. The other interesting, or the other potentially new thing that's come out of this modeling exercise came out of including seed dispersal in this model. So until now, people have only really thought about the effects of the negative effects of clonality on sexual fitness via its effects on mating. But it turns out it might have positive effects on sexual fitness during seed dispersal. So here I'm depicting three outcomes of the model. So this is the relative fitness of different, representing the fitness of plants that have invested in different amounts of clonal growth, the fitness of plants through their male function, through their female function, and the overall fitness of those plants. And I just want to start with this figure here. You can see that intermediate levels of clonal allocation, I should perhaps say, this is the relative fitness of clonal versus non-clonal phenotypes. So values of 1 indicate that clonal phenotypes had higher fitness than non-clonal phenotypes. And you can see that for restricted and intermediate dispersal distances, these red and black curves here, clonal phenotypes tended to be favored, tended to have higher relative fitness than non-clonal phenotypes. This total fitness is just the sum of fitness through female function and male function, so the fitness through pollen and seeds. And you can see that the majority of fitness, this fitness benefit to clonality, comes from female function, not from male function. And so this is coming through post-mating processes, including seed dispersal. So an interesting outcome of this model then, or potentially novel outcome of this model, is that clonality is an evolutionary blessing in terms of Darwinian fitness, in the sense that it might promote the successful dispersal of seeds. It might reduce competition among seeds that have been dispersed, and it enables the dispersal of those seeds over a broader area than in an equivalent non-clonal organism. Okay, so I just want to sum up the major messages of this talk. Okay, so clonality does cause mating interference. It's just that the magnitude of mating interference in these clonal plants is at less than an equivalent sized non-clonal organism. So the models support this idea in the literature that clonal interference occurs. It's just that maybe it's not as important as we've thought before. The major fitness effects of clonality appear to occur after mating, particularly during seed dispersal. And this is a new thing that comes out of including seed dispersal in these models. And the other thing that had not been discussed before in the literature is that by including different amounts of different average dispersal distances in the model, and by showing that clonality is more strongly favored when dispersal is restricted, this suggests that clonality is favored by dispersal limitation, because clonal organisms can by the very nature that they occur over a broader area, can disperse their seeds over a broader area when seeds or pollen are intrinsically limited in their dispersal distances. Okay, so clonal expansion is just one mechanism by which plants have to become larger. The mating costs of this large size are well established. In fact, Spencer Barrett did some of the clearest research on this. He showed that as plants get bigger and bigger, these mating costs increase. But this is not a problem that's unique to clonal plants. In fact, the nature of clonal plants, the spatial subdivision of reproductive function in clonal plants actually reduces this mating interference. And moreover, this spatial subdivision, this spatial expansion might actually promote the fitness of clonal organisms during seed dispersal. Okay, this work was done in collaboration with Wendy Van Drunen, who was a master's student in my lab, and with whom I continue to collaborate on these modeling efforts, and with Mark van Clunen at the University of Constance in Germany. Thanks very much.